So I'll talk about processing and immunogenicity predictions for class 1 epitopes in the IEDB. And yes. So as we just, Edita just showed you, one approach to T-cell epitope predictions is to just go for MHC binding. But there's other factors beyond a peptide's ability to bind MHC that determine if it will actually be recognized by T-cells. And so essentially there's two sets of tools in the IEDB that try to deal with this. One are processing predictions that are essentially dealing with how the peptides get made. And then there's immunogenicity predictions that deal with the T-cell ability to recognize a peptide. So why processing predictions? So this illustrates that I don't draw very well. That's why I have to label my pictures. Imagine this is a virus and this is a mouse. If a virus infects the host, what it does is it goes inside of cells, replicates, and then spreads to other cells within the host. But within these cells, what happens is that actually the virus, alongside with other host proteins, gets degraded into individual peptides, which then can bind MHC. Some of them get presented on the cell surface, and that's where they get recognized by CD8 T cells. If the T cell recognizes a foreign peptide within the cell, it starts to do several things, including potentially killing the cell if it's a cytotoxic T cell, releasing cytokines to attract other immune cells to the site of infection, and ultimately proliferate and form memory to be prepared for a second response like this. So the goal of T-cell epitope mapping is then obviously to identify, given the set of proteins encoded in a virus, what are the epitopes, what are the fragments inside these proteins that are recognized by T cells. And obviously, as we had just talked about, the ability of a peptide to bind to MHC is important, but what other factors play a role inside the cell that determine what peptides get made? So essentially, what happens inside the cell is that you have proteins continuously. All proteins in the cell are subject at some point to degradation by the proteasome, where they get cleaved into peptide fragments somewhere between 8 and 20 amino acids in length. Those get normally degraded, again, to individual amino acids. So you actually have a continuous turnover of proteins being made and degraded into amino acids, which is actually also a way of the cell to kind of to reset its state. But what happens to some of these peptides is that they escape degradation by peptidases and get transported by a transporter called TAP into the ER. In the ER, they are again subject to degradation, this time from the N-terminus only though. And some of the peptides escape that degradation if they are capable of binding to MHC molecules. These bound peptide MHC molecule complexes is what gets then transported through the Golgi onto the cell surface. So here then you now end up with peptide fragments of proteins inside the cell being presented on the surface. And these get scanned by T cells, and T cells during maturation get essentially shown what self-peptides are, so they don't bind self-peptides with high affinity. But if they now come towards a T cell that is circulating in the periphery and recognizes a peptide with high affinity, it knows that this is likely not to be a self-peptide, and it then triggers the T cell response. So again, the question is, given a protein sequence, what is going to be the peptide that triggers this T cell response? And all these factors are specific and select certain peptides over others. But how important are they? We've tried to quantify this, and through a set of experiments, essentially estimate that roughly 1 in 7 peptides can get made and transported into the ER, and that is primarily due to the proteasomal cleavage specificity and the TAP transport preference. And roughly, and this varies per MHC molecule, but up to 1 in 40 peptides can bind MHC with reasonably high affinity and then gets transported to the surface. And then, and this is kind of a tricky estimate, but to the best that we could estimate based on mice immunogenicity studies, out of peptides on the cell surface that bind with high affinity, about 50% can trigger a T cell response. So obviously, this means that the MHC binding is the most selective part of these processes, and that's why we focus so much of our predictions on them, but the others are important as well.
Question, in that model, was it that 1 in 40 of the peptides that make it through TAP, or just 1 in 40 of all?
1 in 40 out of random. Yeah. And actually, that's exactly the key issue. And I mentioned that yesterday. There's overlap here. There's strong overlap in these specificities, especially TAP transports preferentially peptides that have exactly the same kind of motifs that MHC like to bind. So predicting these, there's a lot of overlap due to coevolution, most likely. Okay. Right. So we have 2 sets of tools available in the IEDB. One is the combined predictor, where we are working with tools that have been individually trained to model proteasomal cleavage or TAP transport. And they were trained essentially on in vitro data sets where these processes were characterized. Then we have the same thing done with neural networks in the netctl algorithm. But then we also have some of these trained based on eluded peptides. So they are not really modeling specifically what the proteasome does or what TAP does, but you're looking at the overall motifs that are surrounding peptides that get naturally processed and presented. And there's a second set of tools called MHC-NP, which is also specifically trained on naturally processed peptides. And then we have a separate predictor that specifically deals with TCR reactivity. So I'll start with the first one, the combined predictor. And essentially you get to that on the, I don't know what I'm doing here, on the tools page. When you go to the proteasomal and TAP transport combined predictions. So if you're here, this first part should look very familiar to you. This is the exact same interface that we use for making MHC class 1 binding predictions, because those are essentially, these are part of the prediction algorithm. But in addition to that, you have some novel fields in the bottom, and I'll talk about those now. So essentially, for the proteasome, you have, first of all, the choice what kind of proteasome to model. There's 2 types. There's the immunoproteasome and the constitutive proteasome. And essentially, at the moment inflammation happens, the proteasome switches to the immunoproteasome type. They actually have very similar specificities. Primarily, are actually, when you look at the in vitro digest, the primary difference is speed of cleavage. But so essentially, for the purpose of epitope predictions, we would normally recommend to stick to the immunoproteasome type. Oops. And essentially what the proteasomal cleavage prediction does, it looks, can the C-terminus of the peptide be created by a proteasomal cleavage? We're typically ignoring the N-terminal site because that is normally digested further by proteases. So we do see a clear overlap between the proteasomal cleavage specificity and the C-terminal end of peptides, not for the N-terminal end. Again, the motif was made by evaluating in vitro digests of proteins by the proteasome and essentially looking for the pattern where the enzyme cleaves. As I said, normally we should stick to the immunoproteasome version of this, but if there's a specific reason, you could obviously also check the constitutive one. Secondly, TAP transport, there's 2 parameters here. And what you saw in that diagram was that essentially TAP transports not necessarily the final peptide, but it can also transport an N-terminally extended peptide, which then gets processed further in the ER. So when you want to predict can a given peptide epitope be presented by MHC, you essentially need to predict not just the ability of the peptide itself to be transported by TAP, but also potential N-terminal extensions of that peptide. And we've done that and benchmarked it and ultimately come up with a model by you making multiple of these predictions. And that model, modeling this length distribution, is parameterized by these 2 parameters, which is essentially dealing with how many extensions to take into account and how to weigh them. And unless you've read the paper, I would leave them like they are. This is what we benchmark to be the best performing version of the tool. And yeah, it's somewhat complicated. So I would stick to those. when you now run these predictions, this is the output you're getting. You have, again, as always, the positions of the peptide sequence. And now you have, in addition to the MHC binding predictions, the proteasome score and the TAP score. Here, because of historic reasons, we had different signs. We had like for MHC, you saw that the MHC IC50 value, the lower the better. But for TAP and proteasome, we had scores that are the higher the better. So to make them compatible, we made the MHC score, which is just minus log 10 of the IC50 values. And then you have 3 scores that you can sum up to the total score. And essentially sorting by this total score, where a higher score is better, is what we would use to identify peptides. In addition, you have a processing score, which just adds up the proteasome and the TAP score. That's essentially just, if you don't care about the MHC binding, really just want to know what peptides can be processed, this is what you're getting. What you see actually when you look at the scores is that the MHC one really has the highest variability. So there you have like if you look at the variance, MHC has a variance of 6.7, proteasome of 1.7, TAP of 2.8. So this really reflects, the scores in these methods reflect the selectivity of these processes. So while there's less variation between a peptide's ability to be cleaved by the proteasome, there's a lot of variability in its capacity to be binding a specific MHC. So when we benchmark these things and looked for, okay, how does this compare to do these predictions compared to pure MHC binding predictions? What we have found is that when you look for eluted peptide data sets, so where the ligands have been identified by mass spectrometry, the combined processing predictions outperform pure MHC binding predictions. However, when we look at the ability to predict epitopes, we haven't found a clear improvement of using the processing predictions over the binding predictions themselves. There were some issues with that, which I think this today is no longer that relevant. I made this slide, I think, 2 years ago or 3 years ago, where originally we had not enough real data available necessarily for human epitopes, because in many cases, epitopes were not mapped down to minimal epitopes in humans, or at least not a sufficient number. We want thousands to make a good benchmark data set. Most of the exactly mapped epitopes were done in mice, and mice have different processing apparatus than humans, so we can't use those epitopes to benchmark our performance of our human processing predictions. And the eluted peptides that you're getting by mass spec may be biased to some extent, in the sense that to detect peptides by mass spec, you really have to have quite a sufficient amount in it. So they might be presenting a best case scenario where really everything has to be optimal: proteasomal cleavage, TAP transport, MHC binding, versus for an epitope, you just need a few on the cell surface, or at least not as many as you need in ligand elution studies, so which could explain why we see less of a signal there of the processing. So overall, essentially what we would recommend is, if you have a smaller problem, if you're looking at a virus with 10 proteins, something like that, there's really not much of a need to go beyond MHC binding predictions, and we would just say make all the one peptides that bind MHC, and then just study those. There's not much. The risk of throwing something out with the processing predictions is too high. On the other hand, if you have if you're looking for class 1 peptides in TB, where you have 5,000 proteins or so, we would absolutely recommend to use predictions like this to rationally narrow down further the kinds of peptides to consider. So these kind of predictions, this actually holds throughout this talk, these recommendations for all the tools. There's different variations here, and I'll talk about the variants a little bit. So the first one is the NetShoop, netctl, netctl pan suite of tools, which is, so they are all based on neural networks, and the different tools. NetShop deals specifically with proteasomal cleavage. netctl is similar to the last tool I showed you. It combines NetShop for proteasomal cleavage predictions, TAP transport, and netMHC for the MHC binding predictions. netctl pan just switches out netMHC for netMHC pan. And the key difference of the different methods here is how they use netshop. And this is the page that you come to. Again, you have a very similar interface. And this is the, up top, you select the prediction method. And down here, you select specific the kind of netshop prediction used in the tool. And there are two major kinds. There's the C-term 3.0 prediction, which is based on examining naturally eluted ligands, versus the 20S 3.0 prediction, which is based on in vitro digests. And so while the netshop C-term 3.0 isn't really a proteasomal cleavage predictor, because it's really just looking at the heuristic distribution of ultimate ligands on the surface of MHC, it does work better than the 20S version. So that's why by default, all the tools use the C-term 3.0 version of these predictions. These are a number of the references for these tools. Actually, the tab prediction is shared. The same is used in all these methods. Yeah. Okay. So finally, there's a completely separate MHC natural process peptide prediction. And this is actually was the first example of someone. We have been approaching people for a long time to please submit tools to us to host. And these guys actually won one of the benchmarks performed for predicting naturally eluted peptides. So we asked them if they have interest in hosting their tools on the IEDB side, and they did. So these are, again, predictions specifically for a set of alleles. The coverage isn't universal, but it's important alleles. And the tool otherwise functions exactly like the rest of the tools do. Yeah. Okay, so finally, we have immunogenicity predictions. So for class 1, we do have them. For class 2, the details work on them. We were hoping to have them ready by this workshop, but I guess it's going to be another, I don't know, half a year or so, I would assume. So it has been proven easier to do this for class 1 epitopes than for class 2 epitopes for various reasons. But essentially for class 1 epitopes, what we did was we assembled 2 data sets of peptides from the IEDB in multiple different data sources, where we have peptides that are recognized by T cells and matched peptides that were having the same MHC binding affinity, but were not recognized by T cells. And then we look for the amino acid composition of these peptides while masking the residues that were binding to the anchor positions of MHC. And then we're looking for the frequency of residues that are recognized by T cells in black versus those that are not recognized by T cells in white. And red actually marks how frequent the same residues are in the overall in the source protein from which these peptides were derived. And you see that some residues, like arginines, are just generally disliked in these peptides, meaning that they're bad for MHC binding and bad for T-cell recognition. But then you have a number of residues, tryptophan, phenylalanine, isoleucine, that are significantly higher, more frequent in residues recognized by T cells. Versus you have methionines and lysines, which are actually disliked for T cell recognition, and serine as well. And to some extent, specifically here, the tryptophan, this matches what people have been describing before. And to some extent, the idea is that these, that the side chain of something like the tryptophan, which is quite massive, is more likely to trigger a T cell response than something like an alanine, say. So we can take these scores and now make propensity scores, so meaning these one would have a positive score because it's enriched in the epitopes versus not, and sum them up. And this is done by this tool. So you're entering the peptide sequences in here. By default, by default, the first, second, and C-terminal residues are considered likely anchor residues, and they are masked. If you have this specific allele, you can specify it here, and then that uses the anchor position that we had calculated for those alleles. And we've only validated this tool for 9-mer peptides at this point. But the output then is then simply the peptides and the scores that they get, which are simply the sums of these propensity scores. And the highest score corresponds to a peptide that is more likely to be immunogenic. And this is the results we got from validation data on independent data sets. You see here, this is the T-cell recognition score, and essentially a positive score means it has more residues that are likely to be recognized by T-cells, and a negative score means it has less of the, it has bad residues for recognition. And you can see here that when you look at epitopes, you have an enrichment of the ones that have a positive score. So one way to use this prediction tool is to just ask that a peptide has a positive score. And you see, you will throw out a bunch of peptides that are perfectly fine epitopes this way. So again, if you have a small set of peptides to consider, we would not recommend using these at all and just rely on MHC binding predictions. But if you want to throw out peptides, you roughly throw out half of your peptides if you're asking for having a positive score, and you're throwing out less than half of your epitopes, so you're enriching your peptide set for likely T-cell epitope candidates. Yeah, and with that, I'm done. Any questions?
Sorry, I may have missed it in the last slide, but when you refer to high score being more immunogenic, I missed what was is there a defined cutoff?
Cutoff is 0.
Okay. So
I just meant positive. I should have said just positive.
Mm-hmm.
Yeah. Okay.
Super.
Yes.
Can you please clarify the difference between in the proteasome, the two types, immuno and constitutive, when we will use?
Right. So we would normally always suggest use the immunoproteasome, always. Essentially what happens is in the cell, the constitutive proteasome has, the proteasome is like a complex protein with something like 20 so subunits. And in the case of an inflammation of or if it's in a professional antigen presenting cell, or if inflammation is present, then several subunits get switched over to the immuno subunits. And they have a slightly different cleavage preference, but primarily they are faster, I think, in peptide turnover. So the difference is really small. But based on our evaluations, using the immunoproteasome is better in predicting epitopes. So that's why we would normally suggest using that.
My question is regarding an autoantigen. If we are looking for a patient with autoimmune disease, versus we are not immunizing as external antigens. So it's something that is processed.
Yeah.
So is it better to use a proteasome prediction method rather than the regular MHC binding prediction in an autoimmune patient?
Everything I said, I think, holds for, we're always using infectious disease as the example here, but the same rules should apply for autoantigens just as well. I mean, the MHC binding still is going to be the dominant factor. In the case of autoimmune disease, you'll have inflammation at the site of epitope recognition. You should have immunoproteasome making it. I think all these things should still hold. Obviously, autoimmune disease, there's all kinds of things being wrong. There might be some other factors that are much more important in determining what gets recognized ultimately. But in terms of how to use the predictions, I don't think we would make any different recommendations for autoantigens than for antigens from an infectious agent.
So you said that for immunogenicity prediction tool, the higher a score that you have, it means that it is more, the peptide is more immunogenic. Does it mean that if we give a set of peptides and then rank them by the score, it almost matches the data we get, the immunodominance hierarchy we get from an alias spot?
Well, that's kind of, this slide is trying to illustrate that. So first, yes, but it's not perfect at all, right? I mean, here you have, these are positive epitopes, and here you have an example of a peptide that scores very, very bad think it's the worst overall, and it does get recognized. So there's not a perfect match.
Yeah.
But in general, the trend is, yes, you'll have a higher score for the peptides that are recognized based on something like ELISpot data. This is actually ELISpot data. Yeah.
But it's not perfect. By not perfect, you mean like you have to we have to expect like 90%, 80%. Can you give us a percentage?
Okay, so do I have percentages here? No. If you look here, I mean, first of all, roughly half of your peptides are going to be having a positive score, and half are going to have a negative score. Of your epitopes, I should have these numbers, but I don't. Maybe 75, 80% have a positive score, and 20% have a negative score. So you see a separation, but there's plenty of peptides that are not recognized with very high scores. And there are some epitopes that have negative scores. So it's definitely not perfect, yes.
Following up on this, I would expect your distribution to be very different for the alleles that are better characterized versus not so well characterized in terms of how many are epitopes, not epitopes, and predicted, particularly predicted correctly or not predicted correctly. So would you recommend changing your how many you throw out versus how many you keep based on the frequency of the allele?
Okay, so first of all, this is completely allele independent. This is just the TCR profile. If I would plot this, everything you said is perfectly correct for MHC binding predictions, right? So this is completely allele independent. That's actually also kind of nice, as in you can always use this. You don't need to decide what allele you care about. But everything you said for MHC binding predictions is accurate. Yes, in general, the better the allele is, the better it is characterized, and that tends to go with allele frequency, because the more common alleles have been better characterized, and then there's more data for them in the database, and the predictions are better. But overall, for class 1 binding, really, I think the algorithms are quite mature, and I would not worry too much about it. I think there's much more of a worry for class 2, specifically for DP and DQ alleles, which tend to have been neglected for a long time.